I think it was divine correction of my path that I went exactly where I was supposed to go to end up where I am now. I know the stories, you'd have to have a whole series to hear the stories of divine correction that got me at Baylor as the undergrad program director of biology, because that was not on the card. Like, that wasn't on the bingo card. That was never a thought in my head at all. So my name is Dana D. Dean. I'm from the Gulf Coast of Texas, born and raised. We were rice farmers back. I was always the sciencey one of the family. So all the hunting and all the stuff that happened, that's really got me excited to dig through some. It was creepy, everybody thought as a kid, but I really enjoyed it. So I was the creepy kind of bio kid from the beginning, from the jump, and my sisters would stand off just judging me, right? And so I went to A&M University, and I actually got my Bachelor of Science in Psychology, and then I got a Master of Science and a PhD. So the name of my department, it was through the vet school, but I actually did neuroimmunology, and the whole time I studied an animal model of multiple sclerosis. And so after I got my PhD, I went to NIH for 4 years and did a postdoc there with a gentleman that he was one of the ones that was actually on the first papers that the first line treatment ever for multiple sclerosis was approved by the FDA in 1991. And Dr. Frank was actually on those papers. He was the MRI specialist. And so that's who I just really wanted to go work for, to do more multiple sclerosis research. And I actually got into traumatic brain injury research while I was a postdoc. So when I was actually a freshman at A&M, like I said, I was a psych major, but I didn't want to do so much like counseling, psychology, psychiatry part of it so much. What I really was fascinated with was the brain, obviously, and I was more interested in like the whole premise of like mental illnesses I was interested in, but I was especially interested in like brain physiology and how drug addiction and things like that actually worked. So I was more of a physiological psychology than I ever was more of a social psychology type of direction. But by the time I finished my undergrad education, I'd worked in a chemistry lab, and it was a bio-organic chemistry lab, and I did all kind of lab stuff there. And then I transferred and went to the soil and crop science, because it's the A and A&M. I went to a soil and crop science lab, and I did all types of cell culture, tissue culture there. And so then I really got into that. So just slowly but surely, I say it's divine correction of my path to get me away. I never even looked back in the psychology area, but I really still fixated on the brain as my actual organ of choice for diseases and such. And so when I graduated, I got a job at the vet school as a histotechnology technician, doing all the slides for a professor. And then I found a professor there that had a viral mouse model of multiple sclerosis. And I'm like, that's where I'm getting my PhD. That's where I'm going to get my grad degrees. Students. Students are my favorite thing. I'm glad that we're a Christian university and I can pray for y'all and do all these things and bring out my Christianity, and I'm not in trouble for that. It's encouraged, it's expected. And then also just my coworkers, the faculty and the staff that I work with. Also, I have the Christian feel of all of them, that we're always praying for each other, supporting each other, students that have a great path and just go through with nothing that shakes them, or there's no problem or challenges or issues along the way. That's fine and dandy, but the majority of students don't have that kind of a path. And so I think it's really great that you can sit down with a student, and we know that words matter. You can say words that can crush a student that you never even thought about. It was very flippant, right? Or you can say words that encourage a student, bolster a student, and give them that oomph to just keep going, keep going. So you always have to be mindful of what words you're using and the interactions you have. And the students are worth everything. None of us would be here without the students. We would all just be sitting around chit-chatting about science or something, right? If there weren't students inspiring us to go, 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 go and help. I'm terrible. I've really gotten out of the literature and everything, but now there's so much. So I teach immunology, and there's so many antibody treatments now that are doing so many amazing things. I don't know what I would go fix or target, but I really think I would like to get into antibody treatment, that kind of stuff, because it's fascinating. Like the things we can do now with the genome to make the antibodies we want to make. So we have a student that just graduated and she's already made her first antibody like in a couple of months. Like, are you kidding me? Back in the day when I was in grad school, that would have taken, I don't know, months and months and months, maybe years, and all of that. And that poof. And so that's really exciting to me. And now there's so many different antibodies. So for so many different disease and pathologic disorders that I find it amazing. Like that's one of the application assignments in immunology class. As you go out there, you find an antibody and you tell me what they're doing with it, because it's pretty amazing. See, what I really want to do, I want to have like kind of a histotechnology class. So of course I teach histo. So you have your microscope, you have all the slides made, and you learn cells, tissues, and organs form under the microscope in the transmission electron micrographs, all those kinds of ways. And then form equals the physiologic function. And it's a very, very powerful class for anybody going into anything, whether it's research or any of the medical healthcare fields. So it's something you would use every day. So when you say skeletal muscle to me, I don't see what Dr. Parizzi shows you in the gross lab. I see under the microscope what skeletal muscle looks like, or under the electron microscope what it looks like. And so that's the thing, is I can do that class, but the class before it, that's an amazing career, is the histotechnology that actually makes the slides. And so it wouldn't take a ton of equipment or anything like that. And it would be a really great, more kind of a 3v9R, or maybe I would have a 3100 special topics class where I taught you the actual basics of it as a 1 hour course. And then you go into 3v9R and we could take tissues and make slides from the very, very beginning of cutting the piece, going through the entire processing of it, then bringing it out, sectioning it, putting on slides, then do all the staining. And you can do all kinds of stains and really get into all of that different technology. Students would love it. Students would love it. And then the professors that have labs would love it, because they would have trained professionals basically to come into their labs and really, really support their labs. So if ever asked, that would be so what I want to do. Oh, I want too bad. I always thought the best skill set I had is I was autonomous and I was a loner. I could totally be alone in the lab for 3 days. I'm like, whatever. You know what I mean? Some people really need constant social. So like even in the lab, they want other people there in the lab. They prefer to have other people on a project with them. So they're never really in isolation. You know what I mean? So if you can't do any isolation, research is going to be hard. It's just you, the microscope, and your slides, your samples, right? It's you in the cell culture room and your cells and your treatments and your data and whatever machinery or instruments or whatever you're using. And so that's the thing I think is a really unexpected part of research for a lot of students. They think it's a very social thing, and I think it's more of an autonomous It can be a very isolating kind of thing. And then the other part of it as well is research. It's for a reason. Rarely do you go in and everything works or your hypothesis was correct. And so you're going back and retooling things, finessing things and all that. So the labs you have in classrooms, like OChem or anatomy, of course it always works. It's been set up to not fail, right? And so then when you get in an actual PI's lab and it's research and you're the pioneer and there might not be much literature, there might not be many publications that tell you do this. You might be striking out on your own as a pioneer. And so that kind of stuff can take a lot of finessing. It could take you 9 months, a year, and you never really made much progress. It's possible. And so I think that is a shock a lot of times to students as well, when you're doing certain fields of research that like, I'm not getting anything. I've been doing this for 2 years, right? They think you walk in, you get a paper and you get out. And so, yeah, it just depends. It really depends on the research and what part of it you're doing. And I think it's a shock. Somewhere between something spicy or something cold, because I like cold too. What if it was like chili lime ice cream or something? You know, like make something new. Yeah, it would definitely have to be like spicy but cold and sweet but salty. You know what I mean? Like the combo deal. That's definitely, I would have to, maybe like kettle corn, right? Kettle corn with a little spice on it. That's my final answer. Kettle corn with spice. So my biggest talk I ever give, like if MSO or any of the organizations call me in, I always give a very standard lecture. And that is, I know the path you want and that you're building towards, but you don't know what divine corrections are going to happen along the way. And that is the only way I got here. Like the only way. I've gotten jobs I never applied for. But I didn't get jobs I did apply for, right? And so that's the thing is you believe in yourself and you keep going and you do all the things you should do. Like you can't just sit there and wait for somebody to call, right? But you keep going and driving. But when you get a no or something like that happens and you think it's this huge negative event, think of it as a divine correction thing. It's leading to something so much more amazing than where you thought you were going or what you thought you were going to be doing. And so that's the hardest thing, I think, to embrace when you're younger, is it's all going to work out, right? There's going to be divine correction. How did Dana Dean end up as undergrad program director of the biology department of Baylor? Are you sure that's where I'm going to end up? Because I don't think so. Like, right? It's an amazing thing. And so you just keep moving forward and making progress and do your due diligence. But when there's divine corrections, say thank you because you know something better is coming, right? Yeah, that's the hardest lesson to learn. But the sooner you learn it, the better you'll be. Life will be way more joyful and more thankful. Current biology students, we are so happy you're here with us. You reward us every day. You pepper us with questions. You keep us inspired. We hope we're inspiring you. If we're not, well, tell us, we'll try harder, right? Like if there's something you want to know about or learn about, go sit with your professors, ask them, right? So we're always open to that kind of interactions. And for prospective students, I hope I see you at recruiting events, and I hope I see you in August in seats at our welcome event, because that's the thing, is I think Baylor Biology is really a remarkable program for students, and it doesn't matter where you're going next, whether it's more of a graduate area or a professional medical area, or even if you want to go into industry, right? If you want to go be at a biotechnology type of company, if you want to go do histotechnology in some path lab, wherever you want to go, whatever you want to do, biology can really set you up for success.